Hi dear students, so in today's session, let's see the microbial production of acetic acid. Acetic acid is systematically known as ethanoic acid. It is a colorless liquid organic compound. It has a pungent smell like vinegar. It's a principal constituent of vinegar. The first vinegar was spoiled wine. Glacial acetic acid is the purest form of acetic acid. Its melting point is between 60 to 70 degrees centigrade and boiling point is 118 degrees centigrade. Glacial acetic acid is highly corrosive to metals and soluble in alcohol. It is miscible with water, glycerol, ether, acetone and benzene and insoluble in carbon disulfide. Microorganisms involved in production. Acetic acid bacteria are commonly used for the production of acetic acid. The three important genres are genus Acetobacterium, and the commonly used species are Acetobacterium aceti, Acetobacterium xylinum, and the genus Clostridium. The commonly used species is Clostridium thermoaceticum, which is an anaerobic thermophile. And the next one is the acetic acid bacteria, in which the commonly used ones are Acetobacter and Glucanobacter. The acetic acid bacteria should have following properties. Tolerate high concentration of acetic acid, should require small amount of nutrients, not over oxidize the acetic acid form. It should be high yielding in producing acetic acid. Substances which are commonly used are ethanol, wine and whey, molasses, fermented grains, malt and rice. Acetic acid is produced in following four steps. First is the conversion of starch to sugar by using amylases enzymes. Then there is anaerobic conversion of sugar to ethanol by yeast fermentation. Then the conversion of ethanol to hydrated acetaldehyde. And finally, by dehydrogenation, acetic acid is produced by using aldehyde dehydrogenase enzyme. Methods for industrial production of acetic acid. First one is Orleans method. Then the surface fermentation, that is tripling generation method, and finally the submerged fermentation method. Let's see the first method, that is the Orleans method. It is the old and traditional method. Slow but continuous process. High grade vinegar is used as starter culture. Fermentation is done in large containers and it is filled to 2/3 of its capacity. Wooden containers are laid down on their sides and holes are drilled at top and end sides of the barrel. Then ethanol is poured into the container using long necked funnels from top. Then add starter high grade vinegar. Ethanol convert into acetic acid within 5 weeks or 35 days. Netting or screens are placed over holes to prevent insects getting into the barrels. The barrels are allowed to sit for several months and the temperature is kept at 21 to 29 degrees centigrade. Samples are taken periodically by inserting spigot into the side holes. When alcohol is converted to acetic acid, it drawn off through spigot. About 15% liquid is left to blend with next batch. Surface fermentation method. It is also known as trickling system or German process. Most commonly used industrial method for production of acetic acid. Surface fermenter or barrel is filled with vinegar moistened beechwood shavings. The separate alcohol is poured from top of container and slowly dripped down through the shaving fillings. Oxygen is allowed to container through two routes. One through holes that have been punched into the sides of vat. Second through perforated bottom of vat. Air is introduced through holes by air compressors. When alcohol reaches to bottom of vat, usually within a span of 3 days, it had been converted into acetic acid. The pH range is between 2.5 to 3.2 and the temperature is kept between 20 to 30 degrees Celsius. 80 to 88% vinegar will be produced by this method. And the advantages of this method is that it is of low cost, ease of control, high acetic acid concentration is obtained and less space requirements. Another one is the submerged method. It is newer, faster and more efficient. For industrial processes, 10 to 18% ethanol and 5 times the nutrients used for submerged fermentation are the starting condition for fermentation. When the concentration of ethanol reaches 15 to 16% of the solution is removed and replaced with fresh substrate containing 10 to 18% ethanol. Production plant is filled with large stainless steel containers called acetators. Acetators are equipped with centrifugal pumps at bottom that will pump air bubbles into tank. As pump stirs alcohol, the nutrient enhances the growth of acetobacter on oxygen bubbles. Heat jackets keep the temperature between 40 to 42 degrees Celsius. In 72 hours, ethanol is converted into acetic acid. The vinegar is piped from acetator to plate and then framed to filtering machine. The stainless steel plates press alcohol through pipe filters to remove any sediment, usually about 3% of total product. The sediment is flushed into drains while filtered vinegar moved toward dilution station. Advantages include high productivity, faster oxidation of ethanol, and low cost due to automation. The isolation, purification, and concentration of final product determine the feasibility of the processes. The first operation is cell separation and cell lysis, and it can be done by cross-flow filtration, nanofiltration, electrodialysis, and these techniques are useful to concentrate the glacial acetic acid. Solvent extraction with distillation is the preferred method for chemically derived acetic acid, whereas freeze concentration is used for vinegar. If the acetate is required in the free acid form, there will be additional cost to convert the salt form produced in the anaerobic fermentation into the free acid form. Liquid extraction has been used to recover acetic acid from the chemical manufacture of cellulose acetate, vinyl acetate, and other acetate products. Acetic acid is used as solvent in different industrial processes, and it is used as an active ingredient in fragrance and perfumes, in production of synthetic fibers and textiles, to make ink and indigo dyes, and in the manufacturing of rubber and plastics. It is used in household cleaning for removing smears and streaks from mirrors, to descale coffee making machine, remove urine odor and pet stain, as a food flavoring, as a condiment to produce pickles, as a flavor enhancer to preserve vegetables, in manufacture of mint sauce and production of rice,